Hi, I'm Dr. Mamari, and I'm going to perform the experiment 1, the first week experiment, which is the qualitative analysis of group 1 and 2 known samples. So the purpose for this experiment is to get familiar with the evidence of the reactions when ions are present. And which ions we are talking about, we have set of ions that are classified as group 1 and group 2. We have lead, silver, copper, and bismuth. Mercury could have been part of group 1, so lead, mercury, and silver as group 1, copper, bismuth, group 2, but we are only testing, based on the lab manual, we are testing the lead and silver, and copper, and bismuth. The experiments that I'm performing here enables you to record your observation for the known sample, because the next lab is about the unknown of group 1 and 2. So your known solution definitely is going to have all 4 of those ions. Your unknown, it could have 1 or 2 or 3 or all 4 of them. So based on the evidence and observation you have for this class, for this experiment that we are performing, you are going to use the evidences and your lab notebook in order to identify your unknown for next class. So if I'm teaching the course, or another professor is teaching the course, they are going to set their expectation for how your lab notebook is supposed to look like and how to organize it, a table for evidences or observation. If you are going to use a table or you are going to use a paragraph format, that's the individual choice, and different faculty, they have different style. But in general, all I have to say, be very careful with the evidences today, because that's your reference for next class or next part of the next lab, when you're trying to identify the unknown. The ion name or the ions present in the unknown will not be given to you, only the evidences will be presented to you. Why I'm performing this experiment? Because you're taking 1046 most likely as a remote learning or remote thing, and because due to pandemic you cannot be in the lab to perform the experiment yourself. So I'm performing the experiment. I will share with you the result of the experiment, and you are going to record in your lab notebook your observation. The lab manual is going to be online on Vital Source, and I want you to purchase your lab manual, because we are not presenting the whole experiment and the theory and background. Everything, you find it in the lab manual. So make sure you purchase your lab manual. It's an ebook from Vital Source, and the recorded data. When you record the data, then you can submit your data sheet to your professor as the assignment into assignment folder. Now you will get the data sheet, but you will not get the experiment, and having a lab manual is mandatory. It's not very expensive. It's not a book, so it's affordable. So make sure you purchase it, because it's needed for your lab techniques. Let me open the experiment so we can look at some of these tables that we are going to use for this experiment today. We have first the purpose of the experiment, which we talked about. We are trying to identify which ions are present, which ions are not present. For the known sample, all 4 of them will be present, and we are trying to find the evidences based on the observation that we have. What is going to indicate that lead is present, or silver is present, or is not present? Because the lack of the evidence, is a proof that is not there for the unknown, for purpose of the unknown. We are going to learn about some of the lab techniques like precipitation, selective precipitation, and other theory that goes with the lecture. You also learn in the lecture about the selective precipitation, like some only certain ions they precipitate when you add chloride, because chlorides are all soluble except for the silver, mercury, and lead. But I was talking about the table that your professor is going to ask you to prepare. This is just a sample table. It only goes to step C1, and if you were using table, you have to continue this to like a E2 step, or as you read the procedure, you are going to find out exactly how long the table is going to be, what should be included. So you have to read the procedure before you complete your table. The observation part is going to be completed in the lab. These steps, the reagents that you're adding is based on the procedure. So you have it in the lab manual. The steps, of course, the name is in the lab manual, and what you're adding is in the lab manual based on the procedure, which step we are dealing with. And then the observation, which you will see, and you record your observation while I'm performing the experiment. And you're going to identify which ions this observation are responsible for. So, for example, if you have a precipitate here, is going to be silver chloride or lead chloride, because copper chloride is not a precipitate in aqueous solution, it dissolves. Or bismuth chloride does not dissolve, so it stays in the aqueous solution form. So when you have a mixture of solid and liquid, you identify which parts, which are the possible compounds that they are in the solid form or they precipitate, and which ones are in the aqueous solution form. Reactions that takes place is explained in the lab manual. I'm not going to go through every single reaction, but that's what you have the lab manual for, to refer to it. So the background theory also, it's explained in detail, and your professor could also hold a section for Blackboard Collaborate, and if they want to explain more about it, it will be explained during the lecture or pre-lab discussions for you. So we have time to finish the actual experiment and not to run the experiment, so you could record your observation properly and in time. So there's a flowchart, and this flowchart is very helpful because it tells you, like, if you have a mixture that you get your sample from this container, you add to the test tube. Okay, you add to the test tube. It's not the group 3, sorry. You're looking for group 1 and 2. So you have the group 1 and 2. You place in the test tube, and then the vertical line, it shows the reagent that you're adding. So in step 1A, if you're adding reagent HCl, is going to form precipitate, but it's not going to change all solid. Part of it, it would be a mixture of solid and liquid. What's in the solid form is a mixture of silver chloride and lead chloride, and what's in the aqueous solution part, you have the copper chloride and the bismuth chloride, or copper ion and bismuth ion, they are in the aqueous solution form. Now, how you separate these two from each other, that's the techniques that you are learning in this lab. We have to centrifuge it. We place the test tube with another test tube in opposite direction, and you are going to see the precipitate settles down. You have clear liquid on top, and decant from the test tube to another test tube to separate the solid from a liquid. Then you look at the reagent. You're adding H2O. When you add H2O to the precipitate, the silver chloride is going to stay in the solid form because it's indicated by S, means solid, insoluble, but lead is going to dissolve. Lead chloride is not stable in hot solution, in hot water, so it's going to dissolve in hot water. It changes to lead ion and chloride ion again. It's not easy to find out because it's colorless, is a white or colorless solution. So you use centrifuge again, you separate the liquid, and to the liquid, if you add chromate ion, it would give you yellow precipitate of lead chromate. So in order to find out you have chromate or not, you start with the mix unknown from the mixture, you add HCl, centrifuge, You have the solid. To the solid, you add hot water. To the liquid portion, we're going to add the chromate ion, and that would be evidence for presence of chromate. Now, how would you prove you have Cl? You're going to follow this line down here for the silver chloride. You add ammonia, it dissolves. You add HNO3, it goes back to solid again. That means it's confirmed that you do have silver ion. I just want to make sure you know how to read this flow chart. It's going to make your life easier. It's going to, even if you memorize this chart for your exam or for your unknown, then you can actually get a lot of help from everything in one place. This is like a picture that it says 1,000 words. So we are going to use the flow chart. You have flow charts for group 3, you have flow charts for group 1 and 2. For the unknown, you don't have the flow chart. You are using the same flow chart that you have for sample, with exceptions that some of those compounds may or may not be there. Okay, after the flow chart, in the last page, we start with the procedure. So let me go ahead and start with the procedure because I want to make sure I have enough time to perform the experiment. And then if you have any questions, if I'm teaching the class, you ask me. If another professor is teaching the class, you could ask your professor about any questions that you might have. Make sure you record your observation carefully, because that would be your good friend in order to identify your unknown. Okay, dealing with strong acids, I have to have gloves, I have to have my safety glasses, clothes and shoes on, and all that geared up. And I'm going to grab a pair of gloves and I come back. Okay, I actually got a box, so I don't have to go back too often. Step 1 of the experiment, we are going to prepare hot water bath. Yes, we did prepare the hot water bath. We are going to get 10 drops of the known solution of group 1 and 2. What does it mean is known? That means that all 4 ions are in there. So we get 10 drops into a test tube. Okay. We have 10 drops of the solution into a test tube. Then we are going to add 4 drops of 6 molar HCl. We add 6 molar HCl. Please pay attention what is happening as I'm adding the HCl. Okay. See, a white precipitate is forming. 3 and 4 drops. So I mix it well. Now I have a white precipitate there mixed with a aqueous solution. It's not all solid. Don't expect the whole thing changes to solid, because it's not going to change to solid. Everything is not going to change to solid. Now to this, I'm going to get another test tube. So to the other test tube, I'm just adding a water. I'm adding water because centrifuge, when you watch the safety video for this class, you will see that to set up centrifuge, you have to make sure it's balanced. So when you balance it, the mass has to be the same. I'm going to get same amount of water in another test tube and place it in opposite direction in a centrifuge. Close it and turn it on. Well, this centrifuge, it goes, increments are in minutes. So if I need like 30 seconds, I just have to time it or approximate for 30 seconds and turn it off. If the centrifuge is not balanced, it's going to start moving around. If it's balanced, it will stay still. So it's going to talk to me and say, I'm not balanced. Or if it's balanced, it's going to behave. So I turn it off after 30 seconds, and I take the test tubes out, follow the second part of the experiment, the second step of the experiment. Okay. I don't want to open the centrifuge until it stops. It's not safe and it's not the right technique. You will learn that in the safety video when you watch it. So until I can actually see that it's stopping, then I can open it and take my samples out. Actually had the students that they have hard time grabbing this stuff out, but okay. See, it was all mixed up, and now you have a solid and you have liquid portion. Without disturbing, on the next step, it says add additional drops of the HCl and see if the new precipitate is forming. Okay, I add 2 extra drops and no new precipitate forms. So this means that everything of the silver and lead already precipitated. In the solution part, there is no silver or lead. If I got additional precipitate, what I do, I have to put it back in the centrifuge and separate, make sure that the white precipitate is settled down completely and only the clear liquid is on top. So no new precipitate forms. What I do at this point, I'm going to take a clean test tube and separate the top layer. The top layer is going to be used later on for copper and bismuth. The test tube with the precipitate is going to be tested for part B, which is separation of lead from silver chloride, lead chloride from silver chloride. And hopefully you know it already, how to separate lead chloride from silver chloride. Both of them are solid at room temperature, but when you hot water, lead chloride goes into solution, silver chloride will stay in the precipitate form. So I am going to add drops of hot water. So we're going to add some hot water, about 10 drops of hot water. I added hot water, the procedure, or you could add cold water and then heat it up, but I added hot water and I'm going to keep it in the hot water bath to make sure that it's going to to make sure that the lead chloride dissolves. Keep it for 1 minute in hot water bath. We're going to keep it for 1 minute. So I time it for 1 minute and keep it in the hot water bath. centrifuge it while it's hot. Why do I have to centrifuge it while it's hot? Because if I don't centrifuge and I let it cool down, then the lead chloride is going to precipitate again. So I'm going to centrifuge it while it's hot. "30-second centrifuge should be sufficient." Okay. We're going to wait until the centrifuge stops before we take it out. I'm going to bring some labels. I'm going to actually label my test tubes before I forget which step I need to use them for. I should be fine, but I don't want you to get the bad habit of not labeling. Well, it doesn't say for which step, but I know it's going to be for part B, for B1. B. I'm going to save this for 2B. Okay. Separate the liquid from solid. Because it's a known solution, I can confidently say that lead should be in the liquid form and silver is in the solid form. If it was unknown, all I can say, there could be lead in the liquid form and solid for silver. So lead in the liquid and solid. This is not confirmed yet, because to confirm it, we have to get a little bit more of a more obvious observation. So to the liquid portion, for this liquid portion, now I'm going to confirm it. I'm going to report which part of the procedure I'm dealing with now, because it's important, and that is a part 1C, confirmation of lead 2+. To confirm the lead 2+, we have to add the chromate ion. Based on the procedure, we are going to add 2 drops of acetic acid, and then we add 5 drops of potassium chromate following the procedure. So 2 drops of acetic acid and 5 drops of potassium chromate. So let's go to fume hood. We keep these in the fume hood because some of them, like high concentration or 6 molar acetic acid, it smells very strong vinegar. So if I add the acetic acid, 2 drops of acetic acid, and add 5 drops of potassium chromate, this is for part C. See what's happening? A yellow color precipitate forms. A yellow color precipitate forms, And that yellow color precipitate is confirming that you do have lead ion. So for your unknown, if you see this color precipitate in part C, that means you do have lead ion. And if you don't see that color precipitate, see, it's cloudy. If you see just the yellow color liquid, that's not the confirmation. That's not the positive test for lead. But if you see yellow color solid precipitate, confirms you have lead in the mixture. So after the 4 ion, we confirmed already 1, and that is lead. I don't need this. I'm going to leave in a separate test tube folder for cleanup later, so I don't get confused what I'm using, and go to the next part, which is part D, or part 1D of the experiment. Why 1? Because we are dealing with group 1 element or ion, that is either silver or lead. And then the step that we are working on is D and E. Okay, so which test tube? What do we have? What do we have for silver at this point? Is the silver chloride in solid form. So silver chloride in solid form, because when we add the hot water, we had some white precipitate left in the test tube, and that is supposed to be the silver chloride at this point. So we're going to add ammonia. What happens when you add ammonia? The manual, it does explain with the chemical reactions why this is going to dissolve. The complex that is forming here is going to be aqueous. The ammonia complex of the ion that is forming is aqueous. So we are adding 6 molar NH3. So what is NH3? NH3 is ammonia, and we all know that ammonia is gas. So when we say 6 molar ammonia, that means it's ammonia in water. And ammonia in water, let me see how many drops before I move. 10 drops. Ammonia in water is ammonium hydroxide. So when you say 6 molar ammonia, that should actually be 6 molar ammonium hydroxide. So it's the same thing. So we're going to add 10 drops. That's 5, 6. Drops. To add ammonia until it dissolves. So okay. Silver and centrifuge. So we are going to centrifuge. The size is not the same. The 2 test tubes, I have to make sure to add more water for the other test tube to make it even. Wait. You centrifuge for 30 seconds. About 30 seconds to 1 minute. Or if I take it out, the 30 second is not a fixed number. If I take it out and it's not clear, the top, the supernatant is not clear, I have to put it back. So that's why it's like qualitative analysis. With the qualitative analysis, we don't have exact measurement. Like when we say drops, that means it's a approximate volume. Because some droppers, they have bigger drop size, the other droppers, they have smaller drop size. I just don't want to be too concerned about like if the 10 drops now is 11 drop, what's going to happen? Probably nothing. But for the titration, when you are using burette and pipette for exact measurement, yes, 2 drops is going to make a difference in concentration of the unknown that you are trying to find. Not trying to find how much we have of each ion here. When we say qualitative analysis, we just want to know if we have anything, any of these ions or not. It's just the presence or absence. Okay, I have the clear solution and a precipitate, so I'm going to, I only need the top solution, the clear solution for my next step. This step is basically step 1D1 and 1D2. So this is the 1D2. The precipitate, I don't need it. It needs to be discarded. So I'm putting on the discard test tube rack. Now, we are in part E1. In part E1, you're adding 6 molar nitric acid to this test tube and about 15 drops. And we want to know if the precipitate forms again or not. If precipitate forms, that means we do have silver, nitric acid. So bring the fluid so you could see I'm adding 1, 2, 3, 4, 5, 6, 7, 8, 9. The solution must change to acidic solution. It must be acidic. A white precipitate form when I added the nitric acid. When I add ammonia, HCl dissolved, I had clear solution. When I added the nitric acid, a white precipitate forms. This is part 1E. And the white precipitate is the confirmation that we do have silver ion. Right now, you might think, yes, we knew that we have silver ion, but this evidence is going to help you, your observation is going to help you to identify if you have silver ion for your unknown or you don't. Okay, you're done with the silver, it goes to other test tube rack. Next, we have 2 more ions that we want to make sure we have or not. We are going to go with the supernatant that we had from part B. Okay. So we take the supernatant that we have from part B, and we are going to, we don't know if there is anything in here, if there is any lead left or not. If there is any lead, we have to remove it because it would interfere with the result of bismuth and copper. Have to remove lead, that is part 2A. So we add sulfuric acid If there is lead sulfate is going to form. So we are going to add 3 drops of sulfuric acid and wait for 1 minute to see if there is any precipitate or not. 3 drops of sulfuric acid. Molar sulfuric acid. 3 drops. 1, 2, and 3. Mix it well. Wait for 1 minute. If there is precipitate, it will form, you will see. If there is no precipitate, there is no precipitate. It will be all clear. So, there is no precipitate, it's all clear. There is no lead. If I had precipitate, I would centrifuge and separate it because I don't want lead sulfate to stay there. So, because it's all clear, it's a clear solution, that means there is no lead sulfate. Now, we are moving to part 2B. Part 2B, and we want to separate the bismuth, bismuth hydroxide. How to separate the bismuth hydroxide? Going to add ammonia until the solution is basic to litmus paper. So I have to make sure I have litmus paper here. Yes, we do. If it's basic, how do we know it's basic? If the solution is basic, it would change the red litmus paper to blue litmus paper. Base for blue, remember? Base for blue. So I'm leaving this out so I can actually use it later. So I'm adding 15 molar ammonia or ammonium hydroxide until is basic. And in order to find out is basic, I'm going to use a dropper, mix it, take a piece, take a drop and add it to the litmus paper. So I'm going to use the 15 molar ammonium hydroxide. You want to add to make sure the solution is basic. So what you watch, see what happens when I add the 15 molar ammonium hydroxide. As I'm adding, you see the top of the test tube is changing color. It's giving me like a blue color precipitate, you see? A blue color precipitate is forming. I have to mix it well. The reason it disappeared because the solution is not basic yet. Because if the solution turns basic, that precipitate is going to form and it will stay. So I have to make sure to add enough that the precipitate stays, or I have the I test the solution with litmus paper to make sure is mix it well. Take a drop, place it on the litmus paper. Okay, the red litmus paper turned blue color. What it means that the solution is basic. And it says after is basic, just to make sure you have added enough, add 3 extra drop. So we're going to add 3 extra drop to make sure. 1, 2, and 3. Okay. We added ammonia until it was basic. We add 3 additional drops of the ammonium hydroxide. The litmus paper that is used, I place it in a watch glass to give like 2 minutes time for this. And then centrifuge it. The counterpart of the test tube that I'm trying to centrifuge, it has to have same mass. So because density for this solid now is greater than the water, I add a little bit more water because the mass actually has to be the same. For a perfect technique, I should place on the balance, on the scale, to see how much it weighs and then make sure that other test tube weighs the same. Or I learned it hard way. If it's not balanced, the centrifuge is not going to be happy and it's going to start moving around. Then I know I made a mistake. The other test tube is not balanced. I have to stop right away. That's the key. Otherwise, it will fall and drop. It will move around and fall. But you have to make sure the centrifugation time is enough for the solid to precipitate completely. Now wait until it stops. I repeat because I'm trying to have a better use of the time, be more efficient, because we don't want to just wait and don't use the time. I'm trying to emphasize on the techniques. Okay, nice deep blue color. That means you most likely 90% you have copper, but we're not sure yet, so we're going to separate as based on the procedure. We have to separate the liquid and save it for later to prove that we have copper. If for your unknown you get like this color, most likely you have copper, but we're saving to confirm it later. I need to, next step, to wash the precipitate. So I'm adding some water to wash the precipitate because we don't want any copper to be present. Wash the precipitate, we mix it well, and we separate the liquid. So I have to wash means you mix with water, you centrifuge and separate. Just discard the washing solution. But this solution you're keeping for the next step. Okay, you're just washing the precipitate, right? So when the centrifuge stops, I open it, and when I open the solutions, I don't need it. I need the precipitate. Okay. This blue color is not good because it interferes with the next part of it. So what I'm going to do, I'm going to discard it. We have this white precipitate uh, that is bismuth, but to confirm it, but it appears light blue. Why it appears light blue instead of the white color? Because it needs one more washing. We try to get all the copper out. So well, make sure if you're using a stirring rod, your stirring rod is clean before each time using. You clean it. You don't trust that someone left it, nobody touched it, especially when you're working with someone else. Place it in the centrifuge. Make sure the other test tube has same amount of water to balance the mass, and centrifuge. When we're working group of 2, I usually ask the students not to waste the time because the lab partner could work on the copper, but since it's only me, I have to finish with one before I can move to the next one. Okay. It looks better. So if I make this basic, it would dissolve. And when it does dissolve, I add tin chloride. If I add tin chloride, tin or SN, it can reduce the bismuth from Bi3+ into 0, and it will give you like black color. We expect to get black color, like, but it's not going to be all black. It's not going to be all black. Don't expect like all black color, because I've seen it in the past that you get more of a gray color. If I see one piece of black there, I say, that's it, there is a bismuth there. So if you get like a black color, that's good. If you don't see black color, you get the gray color with particles of the black in there, that means you have bismuth. Okay. So I have basic solution. The solution is basic, and I added tin chloride, SnCl2. Going to mix it well. Wash the stirrer out. Mix it well. But to make sure the solution is basic, is expected to be basic, because 6 molar NaOH is very strong base. Place it in a watch glass and add 1 drop of the solution. The solution is basic, so I could see it already. I hope that you can see it. You see those black particles in there? Those black particles are bismuth. So if you have the black particles, that means bismuth is present. So that black particles with the white precipitate you have, it can give you a gray color, but this is the evidence. So if you see even 2, 3 of them, but I see a lot right now, that is evidence for presence of bismuth. So make sure you record it. Okay? It would be best for you to have a table ready, or have your lab notebook ready, and as you're watching, you record your observation, because you're not going to remember everything. So you want to make sure record your observation as you are watching the video. And the good thing is that you can replay it often. So the black particles here, that's presence of bismuth. It did settle down and I see the black particles. That's how I can omit the centrifuge. Otherwise, I have to centrifuge it in order to see it. But since I already see it, I don't have to centrifuge. Okay, I'm done with the bismuth part. The only thing I need to confirm it at this point is copper, to make sure that I have the copper confirmed. Okay, part 2D. To the copper solution we had from before, we are going to add acetate ion dropwise until the solution is acidic to litmus. What does it mean? If I take a blue litmus paper, take 1 piece of blue litmus paper, and I place in a watch glass, I'm going to add acetic acid to mix it well and take a drop and add to the litmus paper, and I show you the color because it's a blue litmus paper now. When we add enough acid, it changes to when it's acidic. When we add a drop, it would be changing from blue to pink color. So I need the acetic acid. Just want to make sure it's here. Or I took it there. Acetic acid. Okay. The color changes as I add the as I was adding acid. I could see that the color is changing. So if the color change is complete, that's the good time for me to test it. I don't want to test too often, because if I add one drop and test it, if it's acidic or not, of course it's not acidic. Nothing happened to show me that has been no change, no evidence. But after there's the color change in the solution, I can now add the drops into the watch glass, to the litmus paper that I have in the watch glass. And I could see this is changing color from blue to pink, and the solution is acidic. Okay, when the solution is acidic, you're going to add 2 drops of potassium ferric cyanide and formation of the maroon precipitate. That maroon precipitate has given some trouble to other students in the past, but let's see what's going to happen today, because we usually don't get that maroon color. We get like a beige color, but wait until we see potassium cyanide. So we are going to add the potassium ferric cyanide. Solution first. Okay, potassium ferric cyanide. Ferrous cyanide. Okay, we are going to add a couple drops of this. Okay, it actually formed. So you see that new color, the maroon color, or record the color in a way that you recognize it. Precipitate form because it's cloudy, and that is the new color, the maroon color that you have. So you record this because when you see the unknown, if you have copper, that's the color you're going to see. It may not be exact same color, but very close to this color for your unknown. Okay, we are done with this part of the experiment, part 1 of the experiment, which is the known sample. I can work with the unknown, but that would be the next part. So let me stop the recording. This is going to be uploaded to different experiment. It would be recorded as a separate experiment. I'm going to stop recording at this point. Thank you, and I hope that you record all your observation to finish up your data sheet and submit it as the assignment to the assignment folder. Thank you.